Hello children and welcome to my channel Chemistry by Miss Nidhi Banga. Today we will be talking about biomolecules. Biomolecules, I believe, is the easiest chapter in grade 12 chemistry. Why, you ask? Because biochemistry or biomolecule is information based. If you make notes once, you can easily just review them right before your exam, a night before your exam, or a day before your exam, in 1 hour or 1.5 hours at max. And then you can score 7 on 7 in biochemistry because the questions asked are very direct, simple, straightforward, information-based questions. And at times, the tricky questions that usually come from this chapter are in the form of assertion reason. And I'm here to help you with those. Okay, so let's get started. So biomolecules can be divided into the 4 biomolecules that we have in our system. Carbohydrates, proteins, vitamins, and nucleic acids. In this video, I'll be talking about these two. These two, vitamins and nucleic acids, I'll be taking in the next video, part 2 that is. All right, so let's get started. First is carbohydrates. What are carbohydrates? Carbohydrates, as the name suggests, are hydrates of carbon. Really? Let's see. This is glucose. So is this really following this formula of hydrates of carbon? CX, water, Y. That means there are fixed molecules of water for fixed molecules of carbon, fixed atoms of carbon, that is. So yes, actually glucose is following this formula. Okay. So, okay, it fits. Let's move on to the next molecule. Acetic acid. Is it following this formula? It is for C2H4O2. So if you look at it, it is following this formula. Okay, but is this a carbohydrate? It's not. It's an acid. So even though it's not a carbohydrate, it's following the carbohydrates formula. Next, this carbohydrate, ramenose, it's not following the formula of carbohydrate. So this definition cannot be applied everywhere. So this definition is faulty. We do not use this definition anymore. The definition, the more accurate definition of carbohydrate is this. They are optically active. Optically active because they have at least one chiral carbon. That makes them optically active. Polyhydroxy, aldehydes or ketones. That means they have many hydroxy groups. And the main functional group is either aldehyde or ketone. Or the compounds which produce such units on hydrolysis. That means even the polymers of polyhydroxy aldehydes or ketones can be termed as carbohydrates. So, key points of this definition, they have to be optically active, polyhydroxy aldehydes or ketones, or the compounds which give these units upon hydrolysis. That's your definition. Let's apply this to this question. Assertion. Deoxyribose, that is the sugar present in DNA, is not a carbohydrate. Reason, carbohydrates are hydrates of carbon and follow this formula, CxH2OY. Assertion, is this correct? Deoxyribose is not a carbohydrate. No, it's wrong. Deoxyribose is a carbohydrate even though it's not following this formula. Okay, it's the carbohydrate or sugar present in DNA. Reason, this definition does not hold anymore. Okay, carbohydrates are termed as or are defined as polyhydroxy aldehyde or ketones, which are optically active. So both statements are wrong. Let's change the assertion. Deoxyribose C5H10O4 is a carbohydrate. In that case, assertion would have been correct and reason would still be wrong. Okay, now let's move on to the next part. Carbohydrates. Are they equivalent to sugars? Can all carbohydrates be termed as sugars? No. Only some carbohydrates are sweet in taste and only those are termed as sugars, not all. There are some carbohydrates, especially long chain polymers, they are not sweet in taste, so they cannot be called as sugars. Next we have classification of carbohydrates. Now classification always follows some basis. So based on their behavior upon hydrolysis, they can be divided into 3 categories: monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides are single units. They cannot be further hydrolyzed. Examples, glucose, fructose, ribose, deoxyribose. They are single units. All right. Next, oligosaccharides. They are the ones which yield, point to be noted, 2 to 10 monosaccharides on hydrolysis. This has been a board question, that oligosaccharides are the carbohydrates which upon hydrolysis yield how many monosaccharides? 2 to 10, 3 to 6, 10 to 100, more than 100. So the correct option is 2 to 10 monosaccharides. Depending upon the number of monosaccharides, they can be divided into disaccharides, trisaccharides, tetrasaccharides. You get the idea. Examples include sucrose and maltose. They are disaccharides. Okay. Then we have polysaccharides. As the name suggests, they are made up of many monosaccharides. So upon hydrolysis, they yield a large number of monosaccharides. Examples: starch, cellulose, glycogen. Okay, these are the 3 polysaccharides in your syllabus. So on the basis of hydrolysis, they can be divided into these 3 categories. Moving on. Now this is important. Based on their ability to reduce Tollens reagent or Fehling solution, they can be categorized into 2 categories. One is reducing sugars. They are the sugars which reduce Tollens reagent and Fehling solution. Why they reduce Tollens reagent and Fehling solution? Reducing sugars means we know that carbohydrates are polyhydroxy aldehydes and ketones, right? So aldehydes, we know, can be oxidized to acids. When they get oxidized to acids, they reduce others. And what do they reduce? They reduce the Tollens reagent and Fehling solution. These are the mild oxidizing agent. They get reduced when monosaccharides or carbohydrates or reducing sugars, they get oxidized themselves. Now, important to note that all monosaccharides, be it aldehydes or ketones, they all are reducing sugars. That means they all can reduce Tollens reagent or Fehling solution. That means they all have free aldehydic or ketonic group. Now, question needs to be asked. Aldehydes, we know, are easy to get oxidized. So they can be easily oxidized to acid. And when they get oxidized, they reduce Tollens reagent or Fehling solution. But ketones, we know, are very difficult to oxidize. They usually don't oxidize, they don't reduce Tollens reagent or Fehling solution. Then how come here the carbohydrates containing ketonic group are also reducing sugars? Let's see. So ketosis, that means the carbohydrates containing ketonic group, they actually, in basic aqueous solution, they tautomerize into aldoses. That means they get converted into aldoses. Aldoses are the sugars or carbohydrates containing aldehyde group. Okay, so when they get converted into aldoses, or after they get converted into aldoses, then they get oxidized into acid, and therefore they reduce Tollens reagent or Fehling solution. Let's do one question. Assertion. Fructose is a reducing sugar. Yes, fructose is a reducing sugar because fructose, even though it's a ketone, it is a monosaccharide. That means in basic solution, in aqueous basic solution, actually the reagents that we use here, Tollens reagent and Fehling solutions, they are basic. So in basic aqueous solution, the ketosis would get converted into aldoses and then they would reduce the sugars. So this statement is correct. Reason. Fructose has a ketonic group. This statement is also correct. They're both correct. But is this the reason for fructose to be a reducing sugar? It's not. Fructose is a reducing sugar because it reduces Tollens reagent and Fehling solution. So even though this statement is correct, it is not the reason to this assertion. So your correct answer is B. That means assertion is correct, reason is correct, but reason is not the correct explanation of the assertion. Okay, moving on. What are the other examples of reducing sugars? Maltose and lactose, the 2 disaccharides, these 2 are reducing sugars. We will see how and why they are reducing sugars. Non-reducing sugars are the sugars which don't have free aldehydic or ketonic group, and therefore they cannot reduce Tollens reagent and Fehling solution. Okay. Example of that, the only non-reducing sugar in your syllabus is sucrose. We will see why sucrose is a non-reducing sugar. But this is an important point to remember, that sucrose is a non-reducing sugar. Now there's an assertion reason based on it, but we'll do that a little while later. Till then, let's do this one. All monosaccharides are reducing sugars. The statement is correct. They reduce Fellings and Tollens reagent. Again, correct. And correct explanation of the assertion. So the answer for this is A. Assertion is correct, reason is correct, and reason is the correct explanation for the assertion. Even if it had any monosaccharide or even maltose or lactose, the two disaccharides, even then the statement would be correct. Maltose and lactose are reducing sugars because they reduce Fellings solution and Tollens reagent. Okay, next up is glucose. Glucose is a very important monosaccharide that we will be studying in detail. Glucose is an aldohexose. Aldo because it has aldehyde group. Hexose because it has 6 carbon atoms. Okay. It's also known as dextrose. Molecular formula C6H12O6. This I believe you know already. Now, we will be doing some reactions which are basically structure elucidation of glucose. They basically gives us clues about the structure of glucose. That means if we know the molecular formula only, how do we find out what are the reaction which gives us clues about the structure and how this carbon and hydrogen and oxygen are arranged? Now, from board point of view, from this topic, questions are asked in 2 ways. One, they can ask you directly reactions. Write the products when glucose reacts with the following. Then 2 or 3 reagents would be given. Or it can be asked in a way that write the reaction which suggests the presence of aldehyde group in glucose, or which suggests that glucose has a straight chain, or write the reaction which suggests that glucose contains carbonyl group. Okay, so you have to remember not just a product of the reaction, you would also have to know which reactions tell you what. Okay, so let's get started. First reaction is reaction suggesting that glucose has a straight chain. That means all the 6 carbon atoms are linked in a straight chain. The reaction is when glucose is treated with HI on prolonged heating, it gives us N-hexane. Now this gives us clue that all 6 carbon atoms in glucose are linked in a straight chain. Moving on to the next reaction. Reaction suggesting the presence of carbonyl group. There are 2 reactions suggesting that. First is the reaction with hydroxylamine. Here, oxime is formed. The carbonyl O combines with the H2 of hydroxylamine and forms an oxime. You know that's a typical reaction of carbonyl group. So it suggests that glucose contains a carbonyl group. Similarly, upon reaction with HCN, it gives us cyanohydrin of glucose. That is also typical reaction of carbonyl group. Moving on, the reaction that suggests that there is a aldehyde group in glucose. Now, when glucose is treated with bromine water being a mild oxidizing agent can oxidize only aldehydes to acid, not alcohols. So when glucose was treated with bromine water, we got gluconic acid. Okay, that means there was an aldehyde group which got converted into acid, carboxylic acid. This suggests that there cannot be a ketonic group here because ketone cannot be oxidized by a mild oxidizing agent. Okay, so since bromine water is a mild oxidizing agent, it can only oxidize aldehyde to acid and not ketone. Now, if you are asked to write the product formed when glucose is treated with Tollens reagent or Fellings reagent, this is the product. That means glucose gets converted into gluconic acid when glucose is treated with Tollens reagent or Fellings solution because they too are mild oxidizing agent. And apart from that, if you're treating glucose with Tollens reagent, apart from gluconic acid, you will get Ag, that is silver, in its elemental form. If you are treating glucose with Fellings solution, you would get Cu2O, cuprous oxide, which will give brown PPT. Okay, that's just additional information. Okay, so this reaction suggests that glucose has aldehydic group. Moving on, reaction that suggests that there are 5 hydroxyl groups on 5 different carbon atoms. Okay, now when glucose is treated with acetic anhydride, we get glucose pentaacetate. That means there are 5 acetate group in place of 5 hydroxy groups. Okay, one very commonly asked question from this reaction is, for one molecule or for 1 mole of glucose, how many moles of acetic anhydride are used? The answer is 5. Okay, now if you want to know the reaction that happens is, take any hydroxyl group, this will attack carbonyl carbon, the pi electrons will go up and they will then come down, and these electrons will then get shifted here. So this thing, CH3COO, gets linked with this oxygen here, and this hydrogen will combine with the acetate group here and will form an acetic acid. Okay, so for 1 molecule of glucose, if you take 5 moles of acetic anhydride, you get glucose pentaacetate and 5 acetic acids as byproduct. Okay, this is one thing. Next thing is, how would you prove? We know that there are 5 hydroxyl groups, but how do we know that those 5 are on different or on 5 different carbon atoms? We know that glucose is a stable molecule. Glucose is a stable compound and it has 5 OH groups. So that is a proof that those 5 OH groups cannot be on, those 5 OH groups have to be on 5 different carbons. Why? Because if there are more than 1 OH groups on a carbon, it's a very unstable situation. It loses water and forms carbonyl carbon. Okay, so since glucose is a stable compound, there cannot be more than 1 OH group on 1 carbon. Okay, moving on to the next reaction. Reaction suggesting presence of primary alcoholic group. Now, we saw that glucose upon reaction with mild oxidizing agent gave us gluconic acid. That proved that there was an aldehydic group. Upon reaction with a stronger oxidizing agent, nitric acid is a strong oxidizing agent. Upon reaction with strong oxidizing agent, we get saccharic acid, which basically has 2 carboxylic group. One was obviously aldehyde getting converted into acid. The other should be a primary alcoholic group. Why? Because this could not be converted into acid by a mild oxidizing agent, but it's being converted into acid by a strong oxidizing agent. It strongly suggests that there is a primary alcoholic group. Also, when gluconic acid was treated with nitric acid, then also we got the same product. This reaction is also very oftenly asked. Write the products formed when glucose is treated with nitric acid or when gluconic acid is treated with nitric acid. You get the same product. Now, after all those reactions, the reaction which told us about the aldehydic group, the primary alcoholic group, 5-hydroxy, and carbon being in straight chain, gave us a lot of clues. Apart from that, Fisher conducted some other experiments also, which gave clue about the relative positions of hydroxyl and hydrogen bonds or atoms. Now, those reactions are beyond the scope of your syllabus, so we are not discussing that. We are just discussing what the structure of glucose is after all this. This is the structure of glucose. This is the structure of open chain. This is the open chain structure of glucose. Okay, here it's very easy to remember. You have aldehydic group on the first carbon, then second, third, fourth, fifth, sixth. This is how we count from aldehyde to primary alcohol. And remember it like, I always remember it backwards. These two are obvious. Then I remember OH, OH, H, OH. This sequence I remember, and it helps me a lot. Okay, so this glucose structure is called as D. Then there's a plus sign, glucose. Now, what is the meaning of this D and plus sign? Let's talk about capital D, capital L configuration. Capital D, capital L mean that the lowest asymmetric carbon. Lowest asymmetric carbon means this is symmetric carbon, the lowest carbon, which is the symmetric carbon. Now, this cannot be asymmetric. So this is the lowest asymmetric carbon because it has this group, this group, this group, and this whole thing. 4 different groups. So it's the lowest asymmetric carbon. In the lowest asymmetric carbon, if the OH group is on to the right, then it belongs to D series. If it's on to the left, then it belongs to L series. How do we decide that? We have actually taken some standard molecules, which is glyceraldehyde. We have 2 glyceraldehydes, D plus glyceraldehyde and L minus glyceraldehyde. These we took as standards. So D plus glyceraldehyde, it has OH group onto the right, okay, and it's on to the lowest asymmetric carbon. So any compound where the lowest asymmetric carbon has OH onto the right would be termed as D compound. Any compound where the lowest asymmetric carbon has OH onto the left would be termed as L compound. Okay, so this is the standard reference compound. Now, that was about capital D, capital L. What about this plus and minus sign? This plus and minus sign tell you about how they rotate the plane polarized light. So, if a compound rotates the plane polarized light clockwise or to the right, it would be termed as dextrorotatory or D isomer, small d mind you, or plus isomer. Okay. If it rotates the plane polarized light anti-clockwise or to the left, then we would call it levo, spelling mistake, levo with A first and E next. Levorotatory or small l isomer or minus isomer. Okay, this I'm not taking in too much detail because this is actually part of your haloalkanes and haloarenes chapter. Okay, but important thing to remember here is that this plus or minus, small d, small l, they are the same thing. They tell you how the plane polarized light is rotated. It has nothing to do with capital D, capital L or capital R, capital S. Okay, this tells you conformation. This is something else entirely. Here we rotate the molecule. Okay, we give priority and then we rotate and then we decide whether it's R or S. It's beyond your syllabus, but students generally get confused between RS and DL. Okay, they are not the same. How? By looking at the structure, you can tell whether it's R or S. You can tell whether it's D and L. But small d, small l or plus or minus, or in other words, dextrorotatory and levorotatory, you cannot tell just by looking at the structure. You have to pass it through polarimeter. Then you would see the optical rotation. That will tell you whether it's dextrorotatory or levorotatory. Okay, so plus minus, small d, small l, they are the same thing. But capital D, capital L, small l, not the same thing. Not the same thing. Okay. Examples of that. See, there is a molecule with S configuration and it's dextrorotatory. There is a molecule of S configuration which is levorotatory. Then D-fructose is a compound which belongs to the D series, but it's levorotatory. That means it has small l or minus sign. Okay. And then there is L-alanine, which belongs to the L series. It's dextrorotatory. That means it will get small d or plus sign. Okay, so these are the proofs that there's a significant difference between capital D, capital L and small d, small l. Okay, all right. Now, an assertion reason question on that. Assertion: D plus glucose is dextrorotatory and L minus glucose is levorotatory. Okay. D plus glucose is dextrorotatory. We know that from the plus sign. L minus glucose is levorotatory. We know that from the minus sign. So this statement is correct. Okay. Reason: D compounds, capital D compounds, are always dextrorotatory and L compounds are always levorotatory. No, this is a wrong statement. Capital D compound, capital D notation has nothing to do with dextro or levo. It has to do with the OH position on the lowest asymmetric carbon. Okay, all right. Okay, next up is cyclic structure of glucose. Now, we saw that Glucose gave reaction with HCN to give cyanohydrin of glucose and hydroxylamine to give oxime of glucose. So those are typical reaction of carbonyl group. There are some other typical reactions of carbonyl group which are not shown by glucose. They are reaction with Schiff's reagent or 2,4-DNP, that means 2,4-dinitrophenylhydrazine. They are typical reaction of carbonyl group which are not shown by glucose. Then glucose also doesn't show reaction with sodium bisulfite. Then glucose shows a reaction with hydroxylamine, we saw that, but the pentaacetate of glucose, that does not give a reaction with hydroxylamine. Now why is that? These three reactions, these 3-4 exceptions prove that there is an absence of aldehydic group, free aldehydic group. That means that aldehydic group is bound somewhere, is involved in some sort of linkage. That is why it's not free to react with these reagents. Okay, but how can glucose react with HCN and hydroxylamine but not react with Schiff's reagent, 2,4-DNP and sodium bisulfite? Why is that? That is because HCN and hydroxylamine, they are good nucleophiles. Good nucleophiles means that what happens is glucose actually exists in a cyclic structure, in a cyclic alpha-beta hemiacetal form, which we will see in a while. Glucose exists in that, in which the aldehydic group of glucose is involved in ring formation and therefore is not free to react. But then with HCN and hydroxylamine, it reacts, because with HCN and hydroxylamine, they, being good nucleophile, they open the ring structure and the aldehydic group then becomes free to react with HCN or hydroxylamine. Whereas Schiff's reagent, 2,4-DNP and sodium bisulfite, they are poor nucleophile. They're mild reagent. They're not able to open the ring structure of glucose and therefore they're not able to free up the aldehydic group and therefore they do not give the reaction with glucose. Next question. Why glucose reacts with hydroxylamine but pentaacetate of glucose does not react with hydroxylamine? How hydroxylamine reacts with glucose, that we just saw, that the ring structure opens up into open chain form and then reacts with hydroxylamine to give oxime. But when it's glucose pentaacetate form, then the ring does not open even with hydroxylamine. Therefore, the pentaacetate of glucose does not react with hydroxylamine. Next. Now, what is the cyclic form and how does it form? So, this is your typical open chain structure of glucose. Now, what happens is, it sort of circles, bends, and then this oxygen, the oxygen on C5, which has lone pairs, this attacks the carbonyl carbon being partially positive. Okay, this one attacks here, these pi electron shift here, and this H then combines with the O negative that is formed, and as a result you get OH instead of C double bond O. Now if that OH shifts to the right, then you get alpha D-glucose, and if that OH comes onto the left, then you get beta D-glucose. Okay, and when that happens, as you can see, it now becomes a closed structure with an oxygen in between. Now this can be drawn like this. Here, if you see, this was the first carbon. Upon reaction with this OH, this carbonyl carbon became OH, which came here in this alpha D-glucose. So this is your first carbon, this is your first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. Now, if you have to draw this in a hexagonal form, you draw it like this. This O being here, and then this carbon, the first carbon is here. Okay, so first carbon, second carbon, third, fourth, fifth, and then this is the sixth. Okay, now you remember last time we remembered the sequence of OH as OH, OH, H, OH. The same sequence applies here. Okay, when you draw this, you know that oxygen comes to one corner, top right corner. CH2OH, the sixth carbon comes up, and then it follows the same sequence. OH, OH, H, OH. These four will come down. then obviously HHOHH would be up. And if you remember this structure, it's very easy to remember the beta structure. All that you have to do is reverse this. Okay, that's it. That becomes your beta D glucose. Actually, it becomes your beta D glucopyranose. And this is your alpha D glucopyranose. Why pyranose? These structures are called as pyranose because they resemble the pyrine compound, which is a 6-membered cyclic structure with oxygen as one of the member. Okay, so this skeleton structure resembles this structure. The analogy is same. That is why these cyclic compounds, cyclic structures of glucose are called as pyranose structures. Now, what is the relationship between alpha D glucopyranose and beta D glucopyranose? They are called as anomers. Why? Because they are essentially the same compounds. They only differ at the configuration at C1. Rest everything else is same. So that is the relationship between them. Any cyclic monosaccharides which differ from each other at the configuration of either C1 or C2. C1 specifically when we are talking about aldosis, and C2 when we are talking about ketosis. This we shall see in a while. Next up is fructose. Fructose is a monosaccharide. It's a ketohexose. Ketohexose means the functional group is ketone. Hexose means 6 carbon atoms. The formula is same as that of glucose, C6H12O6. That means it is a functional group isomer of glucose. So the relationship between fructose and glucose is that they are functional group isomers. One is aldose, the other one is ketose. Fructose is your fruit sugar. It's the sugar found in fruits, honey, and some of the vegetables. It belongs to the D series. That means if this is the structure, this is the open chain structure of fructose, the lowest asymmetric carbon has OH on to the right. That is why it belongs to the D series. But it's levorotatory. That means it turns plain polarized light anti-clockwise. That is what levorotatory means. Okay, we show it with small l or minus sign. So it becomes D minus fructose. That would be name of this compound. Now, this also exists in cyclic forms because here also the lone pair present on the 5th carbon would attack the carbonyl carbon. Okay, but here see, now the ring that would be formed would be a 5-membered ring with these two hanging out, and one of the member would be the oxygen. So this is what you get. Alpha D fructopyranose and beta D fructofuranose. Why furanose? Because this is furan. Furan is a 5-membered ring with oxygen as one of the member. And when we draw the cyclic structure of fructose, this also gives us 5-membered ring with oxygen as one of the members. Now here also, see, here the sequence is different but very easy to remember. The alpha D fructofuranose has OH, 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 OH. And beta D fructofuranose, if you remember alpha D, then beta D, you only have to switch at C2. Okay, because this is C1, this is C2. So in case of ketosis, the anomers, that is alpha D fructofuranose and beta D fructofuranose, they differ at configuration at the 2nd carbon. Okay, this is where the difference comes. So they are C2 anomers of fructose. Now very quickly, let's compare glucose and fructose because this too has been a question in boards. Glucose is an aldohexose, fructose is a ketohexose. Glucose is dextrorotatory, fructose is levorotatory. Glucose, the cyclic structure of glucose is called as pyranose structures. Cyclic structure of fructose is called as furanose structures. Then you can also mention in glucose, the alpha and beta are C1 anomers. In fructose, the alpha and beta cyclic forms are C2 anomers. Let's now discuss disaccharides. Disaccharides, as the name suggests, has 2 monosaccharides linked together. There are 3 disaccharides in your syllabus: sucrose, maltose, and lactose. Definition of disaccharides, very easy. It has 2 monosaccharides which are joined by an oxide linkage formed by the loss of water molecule. Now, good thing here is that in boards, it has the structures of sucrose, maltose, and lactose have never been asked. What's been asked is their hydrolysis products or their linkages. Sometimes they give you MCQs where the structure is given. They will give you the structure and you will have to choose the glycosidic linkage between the 2 monosaccharide unit, that is. Okay, so structures you don't have to remember, but you have to remember the hydrolysis product. Another very commonly asked question here is the kind of linkage that exists between the 2 monosaccharides. This oxide linkage is called as glycosidic linkage. It's a kind of ether linkage because it's this kind of linkage, COC. That's why it's also called as oxide linkage or ether linkage, because in ether also you have COC. Okay, so glycosidic linkage, the linkage between 2 monosaccharides when they combine and form disaccharide by a loss of water molecule. Okay, now our first and very important disaccharide is sucrose. Sucrose is your normal table sugar. The formula is C12H22O11. Now if you see, it has just double number of carbon atoms, double number of hydrogen atoms -2, and double number of oxygen atoms -1, double than glucose. So that is because when you hydrolyze sucrose, you get glucose and fructose. Okay, or the formula also makes sense when you think of glucose and fructose combined together -1 water molecule. C6C6C12, H12H12 H24, but -2 hydrogens for water. Then O6O6, that means O12, but -1 oxygen because of the water. So upon hydrolysis, sucrose gives you D-glucose and D-fructose, D plus glucose, D minus fructose. Now this reaction is very important because sucrose as a compound is dextrorotatory. That means if you pass plain polarized light through a solution of sucrose, it will turn it clockwise. But once you hydrolyze sugar or sucrose, then the products that you get, the mixture of the products that you get, it becomes levorotatory. Why? Because it gives you glucose and fructose. And glucose is dextrorotatory by 52.5 degrees and fructose is levorotatory by -92.4 degrees. So this value is bigger. That makes the overall mixture, overall hydrolysis products mixture levorotatory. For this reason, sucrose is also called as invert sugar. And in biological systems, when invert sugar, that is sucrose, is broken into glucose and fructose, it is done so by an enzyme called as invertase. The enzyme which acts upon invert sugar, that is sucrose, in biological systems to give you the same products, that enzyme is called as invertase. Okay. Next, let's talk about the hydrolysis products of the 3 disaccharides that are there in your syllabus. Sucrose, we just saw glucose and fructose, but thing to remember is that it's alpha D glucose and beta D fructose. Maltose, it's very easy. It's alpha D glucose and alpha D glucose. Lactose, it has glucose and galactose. Galactose, from galactose you can remember lactose, or from lactose you can remember galactose. But here they're both beta D. Okay, these things, there's no easy way to say this, you'll have to remember. Okay, now more important than that is in reasoning questions, what's very usually asked is the glycosidic linkage. In sucrose, the linkage that takes place between glucose and fructose is this: C1 of alpha D glucose and C2 of beta D fructose. They combine, and with the loss of water molecule, you get sucrose. Now, We saw the structure of glucose. Remember the open chain structure of glucose? C1 was the carbon containing the aldehyde group. And we also remember the structure of fructose. C2 was the carbon containing ketonic group. Now since C1 and C2, they are both involved in bonding, neither the aldehydic nor the ketonic group is free to reduce the tolerance reagent and Fehling solution. So that makes sucrose a non-reducing sugar because the aldehydic and ketonic groups are tied up, are involved in linkage. Therefore, they are not free to be oxidized. Therefore, they are termed as non-reducing sugar. That means sucrose is termed as non-reducing sugar. Now, let's talk about this assertion reason problem. Assertion: sucrose is a non-reducing sugar. It's true, sucrose is a non-reducing sugar. Reason: sucrose is formed by glycosidic linkage between C1 of alpha glucose and C2 of beta glucose. Reason is false because till here it's right. After that it's beta fructose. So your answer is, assertion is right, reason is wrong. Next we have maltose. Maltose we saw is made by alpha D glucose and alpha D glucose. Here the linkage, glycosidic linkage takes place between C1 of one glucose and C4 of another glucose. So C1, which had the aldehydic group, is busy, but with this glucose unit, C4 is being utilized for linking. That means इस glucose unit का C1 is free. That means the aldehydic group is free. That means it can get oxidized to gluconic acid and in turn reduce tolerance reagent or Fehling solution. That makes maltose a reducing sugar. Next up, lactose. This is the sugar found in milk. Here the linkage is between C1 of galactose and C4 of glucose. Here also this glucose unit has C1 entirely free, C1 being the aldehydic carbon. This carbon is free. That means it can reduce tolerance reagent and Fehling solution. That makes it a reducing sugar. Moving on to the polysaccharides. We have three polysaccharides: starch, cellulose, glycogen. This part, polysaccharides, what is most commonly asked is differences. Differences between these three or any two of them, or differences between amylose and amylopectin, which are the constituent of starch. So let's start with starch, their components. Amylose is something that constitutes only 15 to 20% of starch. That means it's present in less quantity. It is where alpha D glucose units are linked by C1C4. Okay, C1C4 glycosidic linkage. Now C1C4 is a straight linkage. That means it's unbranched. It's the unbranched part of glucose. This is also water soluble. Okay, so these are the four things that you need to know about amylose. Amylopectin, on the other hand, is the major part of starch. It constitutes the major part of starch, and it has regular C1-C4 linkage between alpha units, but apart from that, it also has C1-C6 linkage, which is formed at the branch points. That means it is highly branched part of the starch. Okay, and this is water insoluble. So if you look at starch, starch has amylose or amylopectin. In that, amylopectin is the major part, 85% of it, which has branches. So starch overall is highly branched. It has straight chain part also, but that's rather less as compared to the branched part. Next, we have the three polysaccharides, and we have compared and contrast all of them. Starch, as we know, is found in rice and potato, main sources of starch. So that means it is the main storage polysaccharide in plants. Importance of cellulose is that it's the main constituent, the predominant constituent of cell wall in plant cells. Glycogen, if you know even a little bit of biology, you would know this, that glucose in our body from the food that we eat, which is broken down into glucose, that glucose is stored in glycogen. And this job is performed by insulin. Okay, that is why people who are diabetic, they basically have shortage of insulin or defective insulin, which is why the glucose, the blood sugar level in their body shoots up because it's not being converted into glycogen. So glycogen is the storage because glucose is stored in form of glycogen, right? So it's the storage polysaccharide in animals. Okay, starch on the other hand is storage polysaccharide of plants. Okay, then next point of difference is hydrolysis product. Here in starch, the hydrolysis product, you can write amylose and amylopectin. In cellulose, it's beta D glucose, not alpha D glucose, beta D glucose. In glycogen, it's alpha D glucose. In starch, the amylose amylopectin, they have alpha D glucose. Okay, the linkage is different. We'll be talking about that. But starch also has alpha D glucose. Glycogen also has alpha D glucose. The only thing that has beta D glucose is cellulose. Then, talking about the linkage in amylose and amylopectin. Amylose, we know that it's a straight chain. So the linkage is C1C4. Amylopectin, apart from C1C4, there is C1C6 at branch points. Okay, so these are the linkages here. Here, it's straight chain, no branching. So C1C4. Here, since its structure actually is very much like amylopectin. So it also has C1C4 linkage normally, and at branch point it has C1C6 glycosidic linkage. Okay. Now since this is like amylopectin and amylopectin was branched, this means that glycogen is highly branched. Starch, since it's mostly amylopectin, it's also highly branched. Cellulose, since it has only C1C4 linkage, no C1C6 linkage, that means it's a straight chain polysaccharide. Okay, so I hope you can easily remember them. Starch we know, found in potato and rice, that means storage polysaccharide in plants. Cellulose, since the name has cell in it, you would remember that it's the constituent of plants' cell wall. Glycogen is the storage polysaccharide in animals, in us. Then this is also branched, this is also branched. So here we have C1C4 and C1C6. Here we have only C1C4 because it's straight chain. Then this is alpha D glucose, and this cellulose is the only one which has beta D glucose.